Now, as far as bioassays are concerned, these are the evaluations of pesticides, which can be done under laboratory conditions. We may start with what goes on, especially with insecticides, because a lot of bioassays are done on insecticides. Then we will talk about mostly toxicology. So let me share the slides on the slide on the evaluation of toxicity. I mean, toxicity is an important pesticide characteristic. So what is pesticide toxicity? The toxicity of an insecticide is usually expressed in terms of LD50 or lethal dose. It is the dose per unit weight lethal to 50% of the population of the organism. So LD50 is expressed in milligrams per kilogram. So it is milligrams of the pesticide for every kg of the organism which has been targeted by the pesticide. We also talk of LC50. This is the lethal concentration. It is the concentration of the insecticide in the external media that will kill half of the test population. LC50. Probably this could describe what can happen to fish. Let's say if you put a pesticide under aquatic conditions, we can talk of lethal concentration or LC50. Because we are actually dealing with the environment which has got the pesticide. With LD50, we are actually feeding the pest with the pesticide. LT50, this is the time required to kill 50% of the test population at a certain dose or concentration. But of all these measurements, we mostly rely on LD50. Now, we want to just briefly look at the testing procedures. I'll briefly explain about what really happens, because we will be, this data on LD50s is obtained by conducting some experiments. What we want is to obtain what they call the dose-response relationship. The dose-response relationship is the one which is used to determine toxicity. So it's simply a graph where you have on the x-axis, you have got the pesticide concentration, and on the response, the y-axis, you have got a percentage death of the animals under test. So you can have the animals for testing being divided into random groups, usually 6. One group is used as a control. This may be treated with a solvent only, not with a pesticide. The other groups are treated with the doses, usually in a geometric progression. For example, you can have a 1 milligram per kg weight, 2 milligrams, 4 milligrams, 8 milligrams, and so on. So you can vary the concentrations and feed the animals with those concentrations of a pesticide. It sounds cruel. And then you have to check how many animals are dying so that you get the dose response relationship graph. After 24 or 48 hours, the response is measured and expressed as a percentage of the total group. As I have said, this can be, cruel to animals. The response is usually measured as probably death percentage. One of the formulas which is used was the one adapted from Abbott in 1925 and is still being used to correct mortalities or deaths. It is x minus y divided by x times 100. This will give you the corrected percent mortality, where x is the percent survival in the untreated controls, y is the percent survival in the treated insects, or it could be some animals. So how do you analyze this data when you are actually subjecting animals to pesticides, trying to check how effective it is in killing these animals? You can use the percent mortality. It can be converted to probits. I will explain later what probits are. The dosage is logarithm converted. So you convert your dosage to a logarithm. So you draw a graph where the dosage is now a logarithm and the percent death is now a probit. This is like data transformation. The above two are plotted in a graph and a log dosage probit line is drawn. Normally you get a straight line, and I'm going to indicate these. The LD50 can be estimated from the regression line from these graphs. Probably we need a sample size of 120 test animals, which is ideal. So what I'm simply talking about is experiments which involve the use of these pesticides, which are being evaluated for their toxicity. We want to find how poisonous these pesticides are by conducting some experiments. So we can vary the dosages which we can feed to the animals and observe how the animals are going to die. Take data and draw some graphs. Here I'm going to talk about bioassays, which are done with insects. Let's suppose you want to find the effectiveness of some, probably it could be new insecticides. First, you may just have to do a bioassay or some laboratory experiments to check whether the pesticide is going to kill the insects. So the first bioassay I'm going to talk about is known as the topical application. Now, in topical application, you have an insecticide which is dissolved in a substance known as acetone. Then you are going to use a syringe, a micro syringe, which you take that pesticide dissolved in acetone and administer a known volume of pesticide to the insect. This is a tedious job and it requires too much time. Now, some insects, due to size, are difficult to treat because you will use a micro syringe to administer the insect which is dissolved in acetone. Then you observe what can happen to the insects and start taking data. So when you are using the micro syringe, you are topically applying the pesticide. Another more or less similar method, which is known as the injection method. In the injection method, you take your insecticide, you dissolve it in peanut oil. If you don't have peanut oil, you use propylene. If there's no propylene, you can use glycol, or again, you can use acetone. After dissolving the insecticide, now you can inject the mixture into the body cavity. Unlike the topical application, here you are just aiming at the insect. Probably you are aiming at the cutiny or the cuticle. But here you are aiming in the body cavity. It may be difficult to find the solvent that is not toxic to the insect. If you are using this method, sometimes acetone may kill the insect, not the pesticide, which you don't want. So these are some of the problems with the injection method, is to find a suitable solvent which doesn't interfere with your experiments. Now, some of the insects may not be injected. In fact, it may not be possible to inject them because of their size and behavior. So we have got the topical method of administering insecticides. We have the injection method. The other one, which sounds nice, is the dipping method. With the dipping method, you simply take the insects, using forceps, and simply dip them in the desired insecticide concentration. Probably you put your insecticide in beakers or petri dishes. Then using forceps, you just immerse the insects into those insecticide concentrations. Now, after dipping, you take your insects and place them in the container and now observe them. Try to find whether they are going to die or not. So this is another method. So please remember, we have talked about topical application, injection method, now dipping. We are trying to administer insecticides to the pests, to the insect pests. The other one is the contact method or residual exposure method. Now with this one, you take your pesticide, again dissolve it in acetone and put it into a glass jar, which is rotated so that the insecticide will remain attached to the walls of the jar. The acetone will evaporate, leaving the pesticide on the walls of the jar. You can vary the dosages. Then you can actually put your insects in the contaminated jars and see their response. Or if you don't want to use a jar, you can use what is known as a precision sprayer, which you can apply the pesticide to various petri dishes, which will be containing leaves with probably aphids. Then you can check whether the aphids are being controlled or whether the aphids are dying due to their contact with the pesticide. Here you are not going to inject anything into the insect. You just create a surface with the pesticides and allow your insects to get in contact with the pesticides. So apart from the contact method, you have the fumigant method, which is probably self-explanatory, where you just fumigate the insects. This particular method is suitable for stored products. A fumigant is introduced in a sealed container with insects. Then you make observations of insects which are now dying because of fumigation. And you can take records at different intervals. All these methods will give you an idea about the dosage to use to kill these insects. Now, there is also what is called the feeding method. Now, this is used to test insecticide against things like larvae. The larvae are placed individually in a container and starved for a few hours. Then they are fed individually. You can use a small leaf disc with a pesticide. When the larva has eaten that leaf disc, you can watch after 24 hours, some of the larvae could be dead. Then you count how many larvae have died. So if I go back a bit, so with insects, number one, we use the topical application. We can use the injection method, dipping method, contact method, residual exposure method, fumigant method, feeding method. Now, what I have been talking about apply to insects, insect pests. Now, that kind of data may only help you when you are evaluating the effectiveness of pesticides in killing the insects. You cannot use that data to predict what can happen when that pesticide is accidentally poisoned a human being. Now, if you wanted to see how pesticides could be dangerous to human beings, you have to use some models, some animals which act like models, so that you can predict what can happen to humans. In these tests, you need rats, mice, rabbits, dogs, and monkeys. However, the number of available animals may limit your studies. But at least what information you get from these animals give you an estimate of how dangerous the pesticide could be to human beings. Well, this is somebody in a laboratory conducting such studies using animals and the pesticides, trying to find out what can happen to the animals. Now, when we are working with the animals, we are going to do what are known as acute toxicity tests. Now, in acute toxicity tests, the animals are actually fed the pesticide or force feed using a tube. After 24 hours, we check the impact of the pesticide on the animals and record their mortality. We get what is called the median lethal dose. That median lethal dose is actually our LD50. And we can use it to characterize the toxicity of a pesticide. Now, the other test which can be done is to use an albino rabbit, which is about 2 to 3 kgs mass. This albino rabbit can be shaved on the stomach area and on its back. Then you apply the pesticide on the shaved areas as a paste, and this is secured with cotton gauze. Now, after 24 hours, you check mortality, which is referred to as dermal LD50. Because what you are saying is that sometimes the pesticide can penetrate via the skin and cause some toxicity. This is actually dermal toxicity and is measured using LD50. So in that case, you are going to use an albino rabbit, where you have to shave it so that you can directly put the pesticide on its skin and study the behavior of that animal. So far we've tested what is known as acute toxicity if the pesticide is eaten. Then the other toxicity is when the pesticide comes into contact with the animal or with the human being. Then there's also acute inhalation toxicity. This is like pollution now. Now, it is obtained by exposing the animals to the pesticide fumes in a chamber for 4 hours. So you create fumes of the pesticides, you put animals in those fumes. You can do the experiment for 14 days and estimate what is known as leather concentration, LC 50 is obtained. This is by polluting the environment. So acute toxicity results from eating the pesticide. Then dermal LD50 is a result of being in contact with the pesticide via the skin. Then acute inhalation toxicity. This is now due to polluted the atmosphere. Now we want to talk about the subacute toxicity and the chronic toxicity. Now, subacute toxic tests or subacute tests could be done over a period of, say, 90 days. But chronic tests could be done over a period of 2 years. And what people look at are the effects of the pesticide on tissues and organ function. We could evaluate things like carcinogenicity. Is the pesticide going to cause cancer? Or teratogenicity? Is it going to affect the fetus or the fetus? Then mutagenicity, is the pesticide going to affect the genes of the animal? Another issue is that can we have what are known as no effects pesticide levels of the pesticides which do not influence the organisms. Now, if we want to understand the effect of the pesticide in a given population which has been exposed to this pesticide, this can be represented by this normal distribution. For example, we could be interested in the population of rats if we subject them to various pesticide levels. We are going to find that there are certain rats which are highly susceptible. They just need very little concentrations of the pesticide to make them die. So they require low dose. Others may respond to medium dose. So these are the majority. Then there is also an extreme end where even if you use a high dose, you have the resistant individuals. So in a given population, you've got highly susceptible individuals which form a small population. The norm is that a medium dose can affect a lot of rats. Another high dose can be resisted by the rats. Now, if we were collecting data in these experiments, normally we do what is known as Probit Analysis. In Probit Analysis, you plot a graph with a dose which is increasing on the x-axis. Then the response is the death of the rats or the animals under test. So if you draw a graph at almost zero, nothing is happening. You reach a certain stage where death rate will increase, and then it reaches a plateau. And what we define as LD50 is 50% death of the animals when they are subjected to a certain concentration. So we find out how much of the pesticide concentration can cause 50% death to the test animals. This can give us what is known as the LD50. And it is the LD50 which we can use as a measure of the toxicity of a particular pesticide. So I'll get on to explain what is Probit. Yeah, I just want to explain about acute toxicity, which we are interested in. This is high dose in a single exposure. We can get results within days. And this is what gives us LD50s. A chronic dose, we are looking at the exposure of people to low doses. Maybe they work at a factory where they are exposed to pesticides. It could be agricultural environment. So repeated exposure over the years will result in chronic effects of the pesticide. And these effects need to be understood so that we can determine safety when using the pesticides. And there's also what we call subchronic. This is medium dose repeated over a few weeks to some months. We may also want to know what these can do. Now we come to the LD values. Here I want to explain this graph. You can see at the beginning where we have nearly zero, there is no death. But suddenly the death is now increasing. We call this the threshold level. Until it reaches here, then that other end. So the higher the LD50, the less acutely toxic is the pesticide. So if a pesticide has got a high value in terms of LD50, it means it's less toxic. Now, in a generalized dose response curve, we have got 3 distinct regions. No detectable response. This is the no detectable response. We have got increasing linear response. The increasing linear response is this region. Then we have got a plateau maximum response. This is the plateau where we get the maximum response. Now, the lowest dose response that produces measurable response is the threshold. According to our graph, it is this area here, the threshold level. Okay, these are the graphs. The first one is showing variation in, if we vary concentration of the pesticide, this is our death rate or percent mortality. We get this sort of S-shaped graph. This graph. But if we change the x-axis to logarithms of the concentrations, then we change these to probits or probabilities, and we can produce a straight line. And this is what they call Probit analysis. Again, another graph is a result of Probit analysis with an equation here, so that you can predict what the chemical can do. Here we've got log concentration, and here on the x-axis we've got the probits. Now, these studies, because remember, we wanted to get information on toxicity concerning the pesticides. Once we know the toxicity levels, we will be in a position to handle these pesticides safely. And in order to get information of LD50s, we are actually using rats, mice, dogs, and guinea pigs for these studies. And we are using these animals as models so that we can now predict what can happen to people. Because rats and some of these animals probably are models which are nearer to what animals can experience in terms of their metabolism. Sorry. Hello, Prof. And Chiripado. And room office. All right. Sorry about the disturbance. Now, these studies can be affected by, let's say, age. If we're working with insects, all the insects could tolerate the pesticide. This can be affected by sex. For example, female insects are more tolerant compared to male insects. Rearing temperature can influence the tolerance. Food supply, what these animals are feeding, can also influence the tolerance. Population density, overcrowding of insects can lower tolerance. Yeah. Okay. Just sign up. Sorry about the disturbance. Right. So I was talking about the sources of variability in dose response tests. What can really influence some of these studies? I had talked about population density, also light conditions. It may affect the activity of many insects or animals. So the information we get from these animals could be influenced by several factors. But however, the data which we get from them, we can still use it to get guidelines on how to use these pesticides. Later on, I will talk about how we can categorize these pesticides into groups according to their toxicity. Anyway, these are some of the experiments which have to be done. Some of them they look cruel, but nevertheless, if we want to understand about the pesticides, we need to do these studies. So this is it on the evaluation of toxicity. Remember, we can use insect pests if we want to have an idea of what can happen to the insect pests. But we cannot use data from insects to predict what is likely to happen to humans. If you want to predict what can happen to humans, maybe we need to use some rats, dogs, and these other animals. But we have limitations about the number of animals which we can use in these experiments. There are also issues about ethics, whether you can actually rear animals so that you get data from killing them. So this is it. I will have to stop sharing. Hello? Hello, are you there?
Yes, Doc, I'm here.
Yeah, any questions on evaluation of toxicity?
No, none from me.
You could easily follow evaluation of toxicity for insects under laboratory conditions.
Yeah,
You are
something
there.
with
Any questions on this topic?
Same here.
So
Yes.
probably we will not proceed any further because I wanted to, from evaluation of
For me, I don't have
toxicity
questions.
under laboratory conditions, just to get information on how toxic are these substances. We may also want to evaluate their effectiveness on the pests themselves under field conditions. Because data on toxicity may give us information on safety. It may also give us some pointers on how insecticides can work. So we may have to do field studies in order to gain some more information about these pesticides, herbicides, insecticides, and fungicides. So the next stage is to talk about such kind of studies. Then from there on, we may have to go into how we can regulate pesticides, because we will now have derived a lot of information from such experiments. So this is what I'm going to talk next, but not today. You may have to give me a date when we can do it. Hello, Grace.
How is tomorrow in
Tomorrow.
how is tomorrow in the in the afternoon maybe.
You don't want around 10, 11?
I don't think I'll be available at that time. I'll be getting discharged from the hospital. I was in the hospital.
Okay, okay. Or we can do it on Monday if you want.
Monday is still fine, Doc.
It's still fine.
Yeah, Monday is fine.
Yeah, so that we don't give you too much at this.
No, it's okay.
Where is your colleague?
I think he's having network.
Huh?
I think he's having network issues. Econet mazwana iri kunetsa zvisingaita.
Okay, let me stop. Okay.